structure models can be found in the main GPCRdb menu under the Structures section here, Structure Models. On this page, you can find our latest structure models. We have separate sections, one for model, where you can find information about the model itself, a link to the details page of that model, when it was built, activation state, whether this model is a refined structure or not. I will get back to that later. And in the receptor section, you can find a link to the UniProt page of that receptor, a link to the receptor page on GPCRdb for that receptor, then receptor, family, class, and species. And in the template section, we only show links to the receptor and structure for entries that are refined structures. So in our latest release, we provide new structure models for which we used AlphaFold to multistate. And if you click on one of the models that is not a refined one, you can find an example of that here. So on this details page, you can see in this NGL viewer how that model looks like. As I mentioned, we used AlphaFold multistate for creating these models. This is a tool that was developed by Lim Hyo and Michael Fagan. It makes it possible to specify which templates to be used when building the AlphaFold models, so you can specify that way sort of an activation state. So we were able to build active state and inactive state models for every receptor. In this viewer, we color the backbone based on the model confidence, the PLDDT score, defined by AlphaFold. So the blue regions have very high confidence, whereas the red are disordered. You can also find some information here about the model, a link to the AlphaFold multistate publication, a download button where you can select the full model or just the model without the loops. And also note that in the PDB file itself that you download, you can find the GPCRdb generic numbers in the beta factor field of the C alpha atoms, whereas you can also find the AlphaFold BLDDT scores in the B factor field of the C atoms, which is the other carbon atom in the backbone. If we go back to the table here, let's have a look at a refined structure example. So you can see that the page of this looks a bit different because a different pipeline was used to build this. So the idea here is to provide users refined structures. So this is based on the structure, the experimental structure itself, in this case 5HT1B, with the 4IAQ PDB code. The purple part of the model is from the experimental structure, but then where there were missing coordinates, the structure, we filled those in based on the AlphaFold multistate model that we created. So you can see, if we zoom in here a little bit, that ECL3, for example, coordinates from that in the structure were missing, so that was filled in with the AlphaFold model. And also that's the case for parts of ECL2 here. And of course also the both N and C terminus and ICL3. In the PDB file of these refined structures, we only show the GPCRdb generic number in the beta factor field of the C alpha atoms. If you scroll down a bit, you can see this little table where we show some information based on a residue level. So you can see that AlphaFold was used to model these residues. But if we scroll down a bit, you can see that we provide links for the receptor and the structure that was this refined structure is actually based on. Also note that in some residues, the rhodomer, so the side chain, will have AlphaFold written here. Those are usually residue positions where either the side chain was missing from in the experimental structure, or so there were missing coordinates, or it's a mutation, and in this case we revert back to the wild type residue using the AlphaFold model. Also, if you would like to download multiple models at the same time, we have this download button here. You can also align the wild type sequences to create a sequence alignment with that button. And also note that we have an archive button here at the top. So we show our previous releases for the homology models. So from 2016 June to 2022 June, we built homology models with our chimeric homology modeling pipeline, and this 2022 August to October set is the first one that has the new AlphaFold models.